I will stop at about 20 till this morning to give those of you that need to take a second attempt on the mastery quiz time to do that. You should be able to finish it in 15 minutes max and still get to your 10 o'clock class. You have a 10 o'clock class. If you do not need to take the mastery quiz, then when I stop class at 20 till, you're free to come. Okay? So make sure you read the lab for Monday coming up for the Monday session. We'll give you a little intro. We're going to do a little talking about the microscope and things maybe in that session. Okay? And then at the end of the period, you will have your quiz on Chapter 4. Page of notes is acceptable. Okay? So, do cells secrete proteins? They can. Some do more so than others. I mentioned at the end of the period last time that you have cells in your pancreas called the beta islet cells, and they are responsible for producing the hormone insulin. And so those particular polypeptide chains, because insulin's a polypeptide hormone, are made on ribosomes and then ultimately have to get out of the cytoplasm of the cell, be secreted into the surrounding environment, and eventually end up in the circulatory system and be transported around your body. Okay? So it is not uncommon for cells to secrete proteins, but not all proteins that the cell produces are secreted. Some of the proteins just maintain themselves and stay in the cytoplasm where they're produced. That would be true, for example, of the enzymes that are involved in cellular respiration, the enzymes of glycolysis, the enzymes of the Krebs cycle. They produce them and they use them right in the cell. So one of the questions I asked at the very end of the period were, where are these secreted proteins produced? And they're produced by the membrane-bound ribosomes. Free ribosomes are the ones that make proteins or polypeptide chains that stay within the cytoplasm of the cell. In eukaryotic cells, there's a complex group of membranous organelles that are interconnected with one another, either physically interconnected. Remember on the micrograph we saw the nuclear membrane was actually physically connected with the membranes of the rough endoplasmic reticulum. But sometimes these membrane systems that make up this endomembrane system are connected to one another via little transport vesicles. A vesicle basically is a little sac. And it is not uncommon for membranes to pinch off these little sacs, and these little sacs then move through the cytoplasm, and they can either fuse with another cell organelle, and therefore the contents that were inside the sac are added to this organelle that they fuse to, or those little membrane-bound sacs, the vesicles, can actually fuse to the plasma membrane, and then the substances are released from the cell. Does anyone know what that phenomenon is called when the cell releases a substance to the outside? It's called exocytosis. Got it in a couple places. Okay? Well, proteins are produced on ribosomes. And if the proteins are going to be secreted, they're going to be produced on membrane-bound ribosomes, the rough ER. Okay? Where do they go from the rough ER? Well, typically what happens is they are packaged up into these little vesicles, and then the vesicles go to another part of the endomembrane system on their way out of the cell. So ultimately they're going to be released from the cell. But along the way, those secreted proteins go to one of these four organelles. Okay, we can eliminate vesicles already because that's what is the transport mechanism to get from ER to one of those first three. So what's the correct answer? Where do secreted proteins go after they're in the ER? Golgi apparatus, lysosomes, mitochondria. Golgi apparatus, okay? Mitochondria, as we'll see later on, are actually not even part of the endomembrane system. Lysosomes are part of the endomembrane system, but that's not the first place typically that secreted proteins go once they're manufactured on ribosomes. So the Golgi apparatus. So this shows you the nuclear membrane up here, physically connected to the membranes of the rough endoplasmic reticulum. Here's our ribosomes that are studding the surface of that ER, and so that's where the proteins are going to be synthesized. Once the proteins are synthesized, the proteins kind of snake their way into this membranous channel system, and eventually a little sac will bud off of these membranes containing the protein that was made, and it's going to move through the cytoplasm, and one of the likely destinations for that is fusing with the Golgi apparatus. What happens to those proteins in the Golgi apparatus? What is the function of that particular organelle? Some of you had the correct function when you wrote down your list of organelles the other day. What's the purpose of the Golgi apparatus?
The repackaging system.
Yeah, it's a repackaging system. It's a place where the proteins can be further modified. Okay? One of the modifications that very commonly happens to proteins is that they are glycosylated. What does that mean? In the process of glycosylation, sugar molecules are actually added onto the protein. So we've actually got a new kind of molecule now, a sugar-containing protein. Those are called glycoproteins. In some cases, we know very definitively what the purpose of adding those sugars onto the proteins are. In other cases, we're still not quite so sure about what is the significance of having these sugar molecules added. One of the things that the sugars might do is basically to provide a little molecular address tag to that protein that kind of marks it for where it should go next. Should it go now to the plasma membrane and be secreted? Should it go to a vacuole? Should it go to a lysosome? And so in some sense, the Golgi apparatus is considered to kind of be the post office of the cell, where all these little molecular sorting tags are put on the proteins that help to determine the destination of that protein and where it goes next. Antibody molecules are glycoproteins. So in the B lymphocyte, which is the cell of the immune system that produces the antibodies, once it's received the proper signals to start producing that antibody, a foreign threat is detected, then those genes are transcribed. The message moves out into the cytoplasm and is translated on these membrane-bound ribosomes. This protein starts to wend its way through these channels of the endoplasmic reticulum and eventually get packaged up in a little vesicle. And ultimately, that vesicle then will fuse with this membranous system, the Golgi apparatus. as that protein then moves through the channels of the Golgi apparatus, it's exposed to various enzymes that are part of these membrane systems. And one of the things that the enzymes might do is stick some sugar molecules on there and glycosylate that protein. There are other kinds of molecules that can be added to proteins. Lipids can be added to proteins, forming lipoproteins. That would require a different enzyme. But are those enzymes present in the membranes of this Golgi apparatus? Yeah, they're there. So different proteins are treated differently depending upon what their ultimate use is going to be in the cell. So that's kind of what this modification process is all about. And then eventually, those modified proteins are packaged up in a new vesicle. So some of this membrane material pinches off. And then this new vesicle is free to go someplace else. Maybe that is to the plasma membrane, where the vesicle fuses with the membrane, and then the contents are released from the cell. Or maybe that vesicle goes someplace else. We'll talk in just a second about what some possible other destinations are for those vesicles. This is probably a good time to introduce an important concept. And that is the concept of a membrane and membrane fluidity. It's not uncommon for science students to have an incorrect perception of what a cell membrane is really like. And they tend to think of it as kind of like a piece of cellophane. And we contribute to that misconception many times in labs, and we will do a lab where you do some looking at the behavior of molecules moving across artificial membranes using this cellophane-like material called dialysis tubing. Have you ever worked with that before, done experiments with dialysis tubing? It really is kind of like a cellophane material, and you put some stuff inside a dialysis bag, you put it in a different solution outside the container, and then you see what moves back and forth across that membrane. But membranes aren't really static like that. Membranes are very fluid, and the components of the membranes are constantly moving and rearranging themselves. That's got to be the case if membranes are able to pinch off of other membranes and to fuse with other membranes. Pieces of dialysis tubing can't do that. Okay? So we're going to come back and talk about this a little bit later on when we spend a whole section on cell membranes and cellular transport. Okay, so what about vesicles from either the ER or the Golgi that contain proteins that are involved in intercellular digestion? What happens to them? Well, they fuse together to form a new cell organelle. Which of these organelles contains enzymes that are involved in intracellular digestion? Yeah, see several trees. The lysosome. Can lysosomes destroy old, worn-out cell organelles? Is that a part of intracellular digestion? Yep, it is. Do things like mitochondria wear out in a cell? Yeah. So what happens to those old organelles? They're recycled. So, this shows you an electron micrograph of a lysosome that contains pieces of actually two old worn-out cell organelles. A mitochondria and then another cell organelle called a peroxisome. And once this lysosome fuses and surrounds these old worn-out cell organelles, the digestive enzymes inside the lysosome start breaking these organelles down into their component molecules. And then the cell can reuse those again. So here's a lysosome. Here's a little membrane-bound sac containing a damaged mitochondrion. The lysosome fuses with the vesicle, and then digestion occurs. So the cell is really pretty thrifty in terms of trying to recycle the molecules that make up its internal cellular structure. And when the cell organelles have outlived their usefulness, then they're broken down and recycled, and we can use those molecules to make other things. There's another important type of intracellular digestion that takes place within the cytoplasm of the cell. And that has to do with the digestion of large food molecules that the cell takes in. This is a food molecule, or maybe a bacterium, maybe a foreign threat. In the environment of the cell membrane can pinch in, surround that molecule, so now it's packaged into a little vesicle or vacuole. And then if that fuses with the membranes of the lysosome, this large complex molecule can be degraded down into its smaller components that the cell can use in its metabolism. What's this process called where the membrane pinches in and brings things in? That's endocytosis, okay? The opposite of exocytosis. These molecules are too big and too complex to be used directly in the cell's metabolism. Consequently, they are broken down into smaller units that the cell can use. It's not unlike the digestive system of your body, your small intestine. When you eat food, you ingest things like complex carbohydrates or proteins, and those are too big to be transported across the walls of the intestine into your circulatory system. And even if they could get there, you couldn't get them out of your circulatory system in the individual cells. Those molecules are too big. So what do? We have hydrolytic enzymes, digestive enzymes in our intestine. And it breaks your proteins down into amino acids. It breaks your complex carbohydrates down into monosaccharides. And then those can move across the intestinal wall into the bloodstream, be transported through the blood, and then be transported into cells. So this is an intracellular type of digestive process. You may or may not remember from the slide I showed yesterday, but one of the players in the cell immunity process, one of the white blood cells, was a cell called a macrophage. And I showed you a little diagram where the macrophage binds to a T helper cell, which then binds to a B lymphocyte. And once all those are together, the B lymphocyte can start producing antibody. Okay? Well, macrophages are famous for a process called phagocytosis. Phagocytosis literally means cell eating. And so if this were a group of bacterial cells, then this could be a macrophage, and it engulfs those bacterial cells. And then once they're inside of a vacuole, they might still be alive. But when they then fuse with the lysosome, they're exposed to those digestive enzymes. And that's one of the killing mechanisms that the macrophages use to kill those engulfed bacterial cells. So intercellular digestion is pretty important. Malfunctions within a lysosome can cause diseases. True or false? That is indeed true. There are some very complex diseases called lysosomal storage diseases. And ultimately, They're the result of a defect in these lysosomes being able to break down and degrade ingested molecules, things that get inside the cells. These particular diseases are genetic diseases. Ultimately, there are malfunctioning genes that code for digestive proteins that aren't working properly. And so in lysosomal storage diseases, these vesicles and lysosomes can be filled up with undigested molecules. And that can have severe physiological consequences for the cell and in the organism. Have you ever heard of Tay-Sachs disease? Tay-Sachs disease is a lysosomal storage disease. And it results in accumulation of undigested lipids inside of the cell. And that can cause all kinds of physiological and neurological problems. It is associated with people of Eastern European descent, showing it has a genetic component. And the more we study things, the more we find that some of the diseases we've known about for a long time are indeed lysosomal storage diseases. There's a little optional point assignment if you're looking for optional points on the website that allows you to go out and do a little bit of research on a lysosomal storage disease and do a little write-up on it. So you might want to just kind of keep track, if you're interested in optional points, of looking at the website and just kind of seeing what some of the options are available out there. So what kind of digestive enzymes would we tend to find, you think, in the lysosomes? If the lysosomes are going to be breaking down complex organic molecules into their smaller subunits, what are the names of some kind of lysosomal enzymes? What are the four classes of organic molecules that make up living organisms? Carbohydrates? One of the things that can break down carbohydrates into monosaccharides are the enzyme amylase. What are the potential enzymes like you find in these lysosomes? Okay. Proteins need to be broken down. A class of molecules called peptidases can degrade proteins. Lipase, good. What's the fourth type? Fourth group of molecules. Nucleic acids. Those are broken down by nucleases. If it's an RNA nucleic acid, it would be an RNase molecule. If it's a DNA nucleic acid, it would be DNase. All of these are examples of a class of molecules called acid hydrolases. Hydrolases are enzymes that take big molecules and chop them up into smaller pieces. What does the acid refer to? Why do we call these acid hydrolases?
They have to be acidic.
Yeah, when we talk about proteins, we'll see that all enzymes have a pH optimum, a pH where they work best. And we tend to kind of think, since cells typically have a fairly neutral pH, that most proteins would work best in a neutral environment. But what about some of the digestive enzymes that are in your stomach? Do they work best in a neutral environment? No. they're also acid hydrolysis. So these lysosomal enzymes require an acidic environment. Well, the pH of the cytoplasm of the cell is around 7. How in the world do we have an acidic environment inside these little membrane-bound sacs? What would you have to do to maintain that acidic environment? What's characteristic of an acid? It's got a lot of what? What's the definition of pH? The minus log of the hydrogen ion or proton, same thing, concentration. So acidic solutions have a lot of protons. So what these lysosomes actually do is in the membrane, there are little pumps that pump protons into the lysosome and therefore create an acidic environment. Why might that be advantageous? To have these digestive enzymes in this little sac where there's more acid, and these particular enzymes have an acid optimum. They work best in an acidic pH. Do you think of any advantage of that?
If it wasn't acidic, wouldn't they denature?
If it wasn't acidic, they would denature. Yep, that's a big part of the puzzle. And that's kind of funny to think about, a protein denaturing at pH 7. But if you've got an acid optimum, if you move away from that optimum, things start to denature. In general, what would happen if the membrane of this lysosome split open and those digestive enzymes started being released in the cytoplasm? Would that be good? No, we'd start digesting the contents of the cell, right? But what's the pH of the cytoplasm? Fairly neutral. So if these break open and rupture by mistake, then those enzymes will denature before they have a chance to really harm the cell. So in some respects, this can be kind of an adaptation for protecting the cell from damage to the lysosomes. There's one other time, thought I had something here, that it actually might be advantageous for a cell to have its lysosomes burst and for the cell to actually self-destruct. Anybody have any idea when that might be? So you're starting to say something bad.
Cancer.
Okay, situations maybe where the cell is damaged in some form. It is also advantageous sometimes for cells to die during the process of organismal development. What happens to a tadpole as it goes through the developmental process of becoming a frog? Loses its tail. What happens? Tissue actually dies that results in loss of that structure. And so sometimes the process of development of an organism requires that cells systematically and programmatically die at particular key times. So cell death isn't always bad, but oftentimes it is. Okay, let's talk a little bit about vacuoles. Can they be formed by endocytosis? Yeah, we've already seen that. When the membrane pinches in, whatever that membrane pinches in and then surrounds is called a vacuole. Might it store substances the cell will need later? Yeah, they can be storage areas. Can vacuoles be formed by smaller vesicles fusing together to make a bigger storage structure? Yeah. Can they be filled with water? Yes. There's a lot of different types of vacuoles that we can find in cells. Here's a good example, and something that we potentially could see. I never can figure these lights out. There we go. This is a paramecium, and this is something you'll see in lab next week when we look at single-cell organisms. Paramecium have a tendency, because their cytoplasm is hypertonic to the environment, to take on water. And water is constantly rushing into the paramecium from the surrounding environment. What would happen to the paramecium if that just continued? Water just keeps rushing in. It'll burst, okay? Be like hooking a balloon up to a faucet and just filling it with more and more water. At some point in time, the cell membrane would rupture. But paramecium have little water vacuoles in their cytoplasm that absorb this excess water. And then those vacuoles can move to the membrane and then release the water back into the environment again. So these little contractile vacuoles are able to keep the cell intact even though water is constantly entering. It's kind of like having a leaky old boat. And as long as you can bail out water faster than it's coming in, you can stay afloat. There are some drugs that you can give to paramecium that prevent these vacuoles from releasing the water contents. And if you expose paramecium to these drugs, then you'll be able to see them get bigger and bigger and bigger and eventually burst. So vacuoles can play an important role in a phenomenon that we call osmoregulation. We're going to come back and talk about vacuoles a little bit more when we talk about plant cell structure, which will be at the beginning of next week. So we'll come back and talk about plant vacuoles in a little while. Okay, very easy one. Cell organelle is a structure adapted for making ATP during cellular respiration. Which organelle? Obviously, mitochondria. Mitochondria are one of my favorite cell organelles, along with chloroplast. Because they very elegantly demonstrate the relationship of organelle structure and organelle function. The way that most of the ATP is made in the mitochondria is by a fairly complicated chemical process called chemiosmosis. And the thing that makes chemiosmosis work is that a hydrogen ion gradient develops, which basically is the source of energy that's used to make your ATP. Now, hydrogen ion gradient just refers to a situation where we've got an area of high concentration of hydrogen ions one place and an area of low concentration of hydrogen ions another place. A good analogy to this is the analogy of a dam. And in this particular analogy, water will take the place of hydrogen ions. Y'all know how a dam works? You've got a reservoir, a lot of water behind the dam. We've got the dam structure, which holds the water back. And then downstream, we might have a river, for example, where there's less water. Built into the dam are little gates called spillways. And if you open those gates, what's the water going to do? It's going to flow from the higher to the lower level. And as it flows through those gates, there's a little turbine inside those gates. And turbines have blades that spin, and when the blades spin, they generate electricity. Okay? That's kind of the way a mitochondria works. Okay? When we talk about cellular respiration, we'll use this room as an example of mitochondria. So if you walk out in the hallway out there, you're in the cytoplasm. This wall represents the outer membrane of the mitochondria. This group of chairs that circles the perimeter of the room represents an inner membrane. So there's a second membrane. Now the analogy isn't real great because actually this inner membrane is very highly folded. It's not just a straight membrane, but it's really folded. What's folding an adaptation for? Increasing surface area. Okay? Inside the inner membrane is a space called the matrix. Between this inner membrane and the outer membrane is another space. For chemiosmosis to occur, we need a space separated by a membrane from another space. So compartment, membrane, compartment. Compartment, membrane, compartment. In one of these compartments, there's a high concentration of hydrogen ions. In the other compartment, there's a low concentration. So lots of hydrogen ions out here. Not so many in here. Okay. Kind of like our dam. The reservoir, the water below, and the dam. In the membrane are some channel proteins, kind of like your spillway in the dam. And those channel proteins let the hydrogen ions move through them. Normally membranes don't like charged substances going across them. These channel proteins make that possible. Is that going to be a natural tendency for things to move from where there's a lot to where there's a few? Yeah. That's what diffusion is all about, isn't it? Something moving from high concentration to low concentration. So as those protons are moving from here to here through a channel protein, this protein is also an enzyme called an ATPase. And that enzyme helps to make the ATP. That's chemiosmosis. You see how the structure of the mitochondria is directly related to its function? It's organized so it has these compartments and a membrane that separates those compartments and a channel protein and an enzyme located within that membrane. Does it make sense now why it's good to have that membrane folded, have more surface area, more room for channel proteins, more ability to make ATP? Okay, I said I would stop. So those of you that don't need to retake your quiz are good to go.